Today we are going to visit the C. elegans laboratory at the University of Oslo. C. elegans is a nematode, which means that it's an unsegmented roundworm with a long cylindrical body tapered at each end. It lives in the soil, feeding on rotting vegetation, and grows to about 1 millimeter in length. Sidney Brenner established C. elegans as a model organism to study how genes function, and he actually did this shortly after Watson and Crick's proposition of the DNA helix model. 60 years of research have led to many seminal discoveries in the field of developmental biology, identifying the molecular mechanisms behind apoptosis and RNA interference. Pooja Kumari, in the lab of Rafal Ciosk, is going to give us a tour of the lab today. Hi Pooja.
Hi Viola, how are you?
Thanks for having me.
Yeah. So today we are talking about how to grow and maintain C. elegans in the lab. Generally we grow them in these culture plates. This is just agar, and then there's a patch of bacteria which we seed on the plates.
And so is that the food?
That's the food.
Yeah.
So we just seed the bacteria, and then you can put worms on them and they feed on the bacteria. Once the food is over, you can actually cut a piece of agar, transfer to a new plate, and that's how you maintain them in the lab.
So how many worms would you have on a plate this size?
Thousands.
Thousands,
exactly. At the same time, you can grow them in liquid culture in these kind of flasks, smaller, bigger. To perform genetic crosses, in order to increase the frequency of males, you can pick the worms at the fourth larval stage. There are 4 larval stages in the worms, and then after that there is a larval to adult transition. So in the last larval stage, you can heat shock them, and at that point germ cells are forming. Then in the next generation you end up getting more males, and this can be between 2 to 5% of the population.
And so can you tell by looking at them that they're males?
Yes, exactly. Yeah. So when you look at them under the microscope, they are different. So the males usually have a tail fan, which is missing in the hermaphrodite. So the hermaphrodite tail is tapered, whereas the male tail is like a fan kind of a structure. Yeah. And they're also smaller in size as compared to hermaphrodite. So it's very easy to identify. Yeah. Okay, so in order to make the genetic crosses, we use smaller plates so that the worms can find each other, right? And then they have limited space to crawl, and then there is more chances of mating, exactly. So what you do is then pick a single hermaphrodite onto the crossing plate, and then allow it to mate with more number of males, usually 3 to 5, so that the chance of mating is more. And we use this wire, so it is actually beaten at the end so that it's like a spoon. You can't see with your eye, but when you look under the microscope, you can see that. And we use that to pick the worms. So you just sterilize it and then wait for it to cool down.
So that you don't burn them.
Of course. And then you can look at the worms using the microscope and then identify the males and then move them to the new plate. So yeah, you just pick multiple males.
Can I have a look?
Yes, you can have a look.
Crawling around.
Yeah, so you leave them in the incubator, let them mate, and then in the next generation, if you see 50% males and 50% hermaphrodite, that means the cross has worked.
Are there mutants that affect crawling behavior?
Exactly. So there are rollers. Rollers are mutants because of defective collagen genes.
Oh, right.
So actually the cuticle, so the worm is essentially rolling inside the cuticle. Then there are dumpy worms. So these are just shorter in size.
Yeah.
So that's why they're fat and dumpy. Yeah, so these are the two classical examples which you can immediately see, rollers and dumpies. And then these are also used as markers for screening.
Great.
So we're going to keep this in the 20-degree incubator over the weekend, and by Monday we should have the next generation. So
it's an efficient model organism.
Exactly. Very, very time efficient and very easy maintenance. So this is how people keep their stocks, experiments. Okay, so Viola, I'm now going to show you the injector, which we use for making any kind of transgenics or also CRISPR-mediated mutations.
Great.
So yeah, so this is the injection setup, which is hooked up to a inverted microscope. And then here you can place the worm on a slide, and using a needle which is attached to this piece of metal, you can inject the germline.
And so then you inject the adults then?
You inject the adults and you put your mixture of plasmids for either transgenics or CRISPR directly in the gonad.
So Pooja, how long have you been working with C. elegans, and what's unique about this model organism?
Right, so I have been working for now more than 5 years using C. elegans. And one of the unique features of this model organism is the invariant cell lineage. So essentially all adult organisms have specifically the same number of cells which were divided and got specified in the same way. And this cell lineage has been completely established. So this kind of cell lineage allowed people to study processes like developmental timing and cell specification. And in terms of time, it's very, very efficient to work with C. elegans. If you want to do either forward or reverse genetics, it's very easy and rapid to do those kind of experiments. In addition to that, we have a lot of tools for transgenics. So there is an established method for developing single copy transgenics. And then these days, with the use of CRISPR, it's almost trivial to make any kind of mutations, be it substitution, insertion, deletion. You can get your line in matters of months, sometimes even in weeks.
Wow, great.
Yeah.
So would you think of any limitations then?
Yeah, so as I said, simplicity is its biggest strength. It also becomes one of the weakness because the animal doesn't have a lot of defined organs or tissues, like it doesn't have a defined brain, or doesn't have blood. So that becomes one of the weaknesses. Also, the size of the animal is really small, so they grow up to 1 millimeter in length. And because of that, doing any kind of tissue-specific studies, like if you want to do biochemistry or proteomics or genomics in a tissue-specific way, it becomes very, very difficult.
And so what do you work on and the lab here?
Right, so I work in the lab of Professor Rafal Ciosk, and he actually has 2 labs, one here at IBV, and the other one in Institute of Bioorganic Chemistry of Polish Academy of Sciences in Poznan.
Yeah.
So the unifying theme for all the people working in these 2 labs is post-transcriptional gene regulation. We look at RNA stability and translation in context of both physiology and development. So the larger group in the lab focuses on changes in gene expression during cold adaptation, and they use both worms and neuronal cultures to study these aspects.
So since RNA interference was discovered in C. elegans, I wanted to ask you how it works in practice. How do you turn off protein expression in the worm?
So of course, RNA interference was first seen in plants, but then C. elegans research contributed a lot to the studying the pathway and identifying all the components in the pathway, right? So essentially, you clone the gene in a plasmid which has promoters from both sides, so that you make RNA from in both direction, ending up giving you a double-stranded RNA. And this plasmid is transformed in a bacteria, and then you feed the bacteria to C. elegans.
Oh, Oh, literally feeding it.
Literally feeding it, exactly.
Yeah. Fantastic. Thank you so much.
Yeah, sure.
Pooja has given us a comprehensive introduction to C. elegans today, and I have definitely learnt a lot. I think for me the ease of maintenance definitely stands out, and I like the way to knock down gene expression by feeding the worms.